Okay, so today we're going to be talking about the animal cell and the functions and structure, or the structures and functions of those structures. Now, most things are going to be pretty similar from the plant cell, and some of the biggest differences are just what's going to be missing from the plant cell compared to the animal cell. Specifically, animal cells don't have cell walls because there are a lot more specialized cells. In animal cells, we get things like bone cells that give us the structure, whereas plant cells, need to use those cell walls to create structure. There's also no chloroplasts, as you know, animals don't create their own food from sunlight, so those are missing. Otherwise, everything you see is fairly similar. There are a few additions, like the flagellum and the centrioles. The lysosomes play a bigger role, but everything else is again fairly similar to what was in the plant cell and does a fairly similar job. We'll go over those again. Okay, so starting with the nucleus, right in the middle of this whole green structure here. The nucleus, it's just kind of contains all of that vital information. It contains the DNA. The nuclear pore just lets information go in and out whenever it needs to. Any energy that needs to go in, any ATP, any waste that needs to come out, those will go through the nuclear pore. The nuclear envelope, again, is just that barrier that contains the everything that is in the nucleus, the nucleolus, the chromatins, the genetic information. It just keeps it separated from everything else so it stays nice and centralized. And then, as we talked about last week, in mitosis and meiosis, while things are splitting off, it will dissolve and reappear depending on what stage it is in. The chromatin is the genetic information. It contains an organism's genome, or its genes. The genes are within the chromatin are made of, again, what's called deoxyribonucleic acid, which is DNA. And again, we'll talk more about that when we discuss genetics. But, all the cells basically have the same genetic information in an animal specific to that animal. And there are certain genes that would be turned on or turned off based on the function of that cell. So if there's a heart cell versus an eye cell versus a bone cell or a muscle cell, different genes are going to be turned on, as it were, in those cells, and that's what's going to provide the different jobs for each of those cells. Okay, and then the nucleolus, it creates the ribosomes, which we'll talk about in a moment. The ribosomes, then, there are some ribosomes, a lot going in and out of the rough endoplasmic reticulum. The regular ribosomes, they're shown on here as all these little black dots. And then there are some just kind of floating all over the place, the free ribosomes. And they are packets, little containers of RNA and protein, and they just create the proteins. RNA is similar to DNA, but a little bit different. It's not in control of the code of what the cell will become, but it has similar coding. We'll talk more about RNA in the future, but the ribosomes really are important in protein synthesis. So if a protein needs to get made, the RNA is going to have the information on how to do that, and then the ribosome contains the RNA, which allows that to happen, and it contains the material necessary for the proteins. to be made. So the rough endoplasmic reticulum then contains those, and that is where that protein is synthesized, and then they're collected there. They're collected in the endoplasmic reticulum for transport throughout the cell. So wherever they need to go, that then happens in the rough endoplasmic reticulum. And then, since we're talking about traveling, the Golgi vesicles are what allows for things to travel around. They work by packaging things, packaging the proteins, packaging other particles, sometimes called macromolecules for transport. They stack larger vesicles with smaller vesicles, and then those can be moved around. So those are the Golgi vesicles, again, working to package everything so it can be transported around the cell. The lysosomes, since they're right next to our Golgi vesicles, they work in intracellular digestion. If there is some waste product in the cell, the lysosomes, and let's see, there's another part for intracellular digestion. Well, other vacuoles. Remember we talked about vacuoles more in plant cells, that big empty space that holds all that water. They're not as prominent in animal cells, but they are still around, and they help the lysosomes with the digestion. And the lysosomes just kind of float around and encapsulate anything that's not supposed to be there, and then they kind of float over to the side and come into contact with one of these secretory vesicles, and then hormones and waste and all sorts of things can leave the animal cell through those vesicles. So it's kind of just like the door of the cell. Okay. The centrioles we talked about in our discussion of, again, meiosis and mitosis. They are what create those spindle fibers to grab on to the chromatins and the chromosomes and pull them to the two different cells that are being made. The spindle fibers are part of that cytoskeleton that's around. Those are the little fibrous parts that create a structure within the cell. And they all float around in the cytoplasm, or actually the cytosol. The cytosol is just that jelly part. The cytoplasm is kind of everything. But the cytosol is actually what is more of the liquidy part. Okay, so that's the liquidy part that everything really is kind of floating around in. Okay, so then we have the plasma membrane, and that sort of separates everything that separates the cells from each other. So we have, it kind of does the same job as the nuclear envelope. It keeps all of its cytoplasm on one side, and then there's a gap between the two cells, and then there's another plasma membrane and going to a new cell. So it provides not the structure, but that separation, the same separation that we get from the cell wall in plant cells also. The mitochondrion provides the energy for a cell. It creates the ATP. That's where any of that ATP is going to be formed. And it is generally referred to, again, as the powerhouse of the cell, or the power center of the cell. And it has a lot of, it's got two membranes. There's an outer membrane that's fairly smooth, and an inner membrane that has lots of very complicated folds. And on these folds, the sugar, or the food, is combined with oxygen to produce ATP. and that's where the cell gets its energy from. All right, so now we have the peroxisome, and they are membrane-bound packets of oxidative enzymes, and they protect the cell from its own production of toxic hydrogen peroxide. Hydrogen peroxide is the chemical symbol, has H2O2, so we've got 2 hydrogens and 2 oxygens, which as you can see is very similar to water, which of course is just H2O, but that extra oxygen, makes it actually a very toxic chemical. And so the peroxisome is what protects the cell from this buildup of hydrogen peroxide. So it kind of goes around and kind of like digests it. That's what it does in animal cells anyway. In plant cells, it works to convert fatty acids to sugar, like we talked about. But in plant cells, it really works to protect from the hydrogen peroxide. Okay, and so next we're going to talk about that smooth endoplasmic reticulum. And again, just like in the plant cell, the smooth endoplasmic reticulum plays different functions depending on the specific cell type, but it does also have lipid and steroid and hormone synthesis, breakdown of lipid-soluble toxins, particularly in liver cells, and it controls calcium release and muscle cell contraction. So it works with a lot of hormones and lipids, steroids, and hormones. So it's working to kind of keep those chemical messengers. It's working with them to make sure their jobs get done, and it's working with the cell to make sure that it's listening and getting the information it needs. Okay. And then at the bottom we have our flagellum. Now in some animal cells, like the animal cells in humans, each individual cell is highly specialized depending on what chromosomes, what genes are turned on, and so one cell can be a bone cell, and really in and of itself it doesn't do too much that is really all that important individually. It's just part of a whole organism. But there are, of course, our single-celled organisms that just their entire life is just this one cell. They're not connected to anything. They're not part of any bigger organism. And so they just work with these things. Now in more complex creatures, again, we have all these different systems and all these different tissues that work together to supply individual cells with what they need. We've got the respiratory system to make sure that cells get their oxygen and get rid of their carbon dioxide. We've got the circulatory system that moves all of the oxygen and carbon dioxide through the blood around the animal to get to all the cells. We've got the digestive system to make sure that all the nutrients get there and the waste gets taken away. And those things kind of happen a bit in individual cells, like we have the lysosomes that kind of work as the digestive system as it takes away the waste, and the peroxisomes as they take away the hydrogen peroxide. But what individual cells like a paramecium don't have is legs, so they can't walk around and get their food or move to a different place with oxygen. So what they do have is this flagellum, and they use it just kind of like a whip to just kind of whip themselves around, almost like, if you're doing the butterfly stroke in swimming, they just have it and it goes around and it kind of pushes them, because most single-celled organisms live in, not all of them, but most of them live in some sort of atmosphere or some sort of environment that has a lot of liquid that may or may not be water. but generally some liquid, and the flagellum works very nicely for locomotion in a liquid environment. So I believe that covers all the different parts of an animal cell. As you can see, most of them have very similar functions to the functions that they performed in a plant cell, with just a few changes and a few additions, like the bigger purpose of the lysosome, and then the centrioles appear, the flagellum on the outside, lack of chloroplasts or a cell wall, but those are really the main differences. So let me know if you have any questions, and otherwise I will talk to you later. Thanks.